Morning, all. So this lecture is on microbial growth and microbial nutrition. For my CCP students, this is chapter 6 of your textbook. For my Camden County students, this is chapter 7 of your textbook. So this is a pretty easy chapter, and I don't want you guys to spend a whole lot of time on this chapter. This would be a chapter that you sort of read through and kind of move on. There's not a whole lot of meat, per se, to really study in this chapter. But we're going to go over what conditions are required for microbial growth. Now, before we talk about microbial growth, growth is actually one cell dividing into more cells, okay? Dividing into two daughter cells, and then continuing the division process. It's not the cells getting bigger per se, it's more of them dividing. So there are several physical requirements that are required for growth: temperature, pH, osmotic pressure. We'll talk about each of those in a second. There are also various chemical requirements. Because all microbes are carbon-based material, they do need to have a carbon source. Nitrogen, sulfur, phosphorus are also important. Trace elements, oxygen, and various organic growth factors. Other important things would be like trace elements. These would be things that trace element means that it's not in a high enough percentage or value to be listed, but it is still required. Oxygen could play a role. We just got done talking about metabolism, or you will be getting into metabolism talking about aerobic versus anaerobic growth. Oxygen does play a role. And then organic growth factors would be things like vitamins. So looking at temperature, most microorganisms are going to be what we call mesophiles. Mesophiles means that they are moderate temperature loving, and this is anywhere from about 20 degrees Celsius to about 40 degrees Celsius. Most bacteria are going to love 37 degrees Celsius. Most fungi will actually love 35 degrees Celsius. Psychrophiles, these are cold-loving bacteria, so anything lower than 20 degrees Celsius. Thermophiles, these are heat-loving organisms. This would be anything greater than 42, 43 degrees Celsius. If we sort of look at temperatures, if you've ever worked in the food industry, you know that the temperature of what food is kept at is critical. And the reason for this is that if you do have food being kept in that 20 to 40 degree range, it is considered a danger zone. And the reason why it's considered a danger zone is because bacteria can replicate very quickly or duplicate very quickly. When we get to infections of the GI tract, one of the biggest causes of food poisoning in the US is actually contamination of cooked food, meaning you touched cooked food, maybe transferred some stuff to that cooked food, and as a result, the bacteria was growing because that food was kept either at room temperature, which is 27 degrees, or maybe even a more lukewarm temperature of maybe 30 to 35 degrees, and you got rapid growth of the bacteria. So it is very important that, if you're in the food service industry or you're cooking food, that food is either kept above or below, those danger zone temperatures. For pH, most bacteria will grow between 6.5 and 7.5. Molds and yeast are more acidic. They like a pH of 4 to 6. And an acidophile would be an organism that really grows in an acidic environment. One area in the body that we'll talk about that's very acidic is the stomach. And there's actually an organism called Helicobacter pylori, which we'll get to when we get to the GI infections. H. pylori can live and reside in the stomach, which can have a pH of anywhere from 0 to 3. And also, as a result, is able to withstand that very low pH. The vaginal tract would be another area. Typically, the vaginal tract has a pH of about 4 to 4.5. So a lot of the bacteria that live there are slightly acidophilic. Osmotic pressure, this is when you have hypertonic environments. Hypertonic would mean like a salt-rich environment. This will cause plasmolysis due to the high osmotic pressure. So this is one of the reasons why we do not find a lot of different bacteria on the surface of the skin. Despite most bacteria having cell walls, they cannot take these hypertonic environments. And what happens with plasmolysis is if you look in the picture here and you look at the right-hand side, what you'll notice is that you're still getting shriveling up of the cell within the cell wall. So the plasma membrane shriveling up, the cell is shriveling up, but the cell wall is still maintaining the shape of the cell. This does not allow for the bacteria to grow, and it may even kill the bacteria. Again, the surface of the skin, very salty environment. We are starting infections of the skin this week as well, so you'll see a little bit more about that. And when you look at the surface of the skin, for example, because of that very high salt environment, the bacteria are not able to grow. But there is an organism that is, which is called Staph aureus. Chemical requirements. These are things that have to, because most bacteria are going to be heterotrophs, which means they have to eat food, they need a carbon source. Carbon is important because it's a structural backbone of all organic molecules. Autotrophs are able to take CO2 out of the atmosphere. Most heterotrophs cannot do that. They have to bring in some type of carbon source. Autotrophs are able to take CO2, undergo a process called photosynthesis, and use that to make carbohydrate or carbon sources, but most bacteria cannot do that process. Nitrogen is very important for cells because it's a component of proteins, amino acids, the amine group, DNA, and ATP. Most bacteria will decompose protein material from nitrogen source. Some bacteria will use ammonia or nitrate from organic materials. And despite the atmosphere being approximately 72% nitrogen, it cannot take nitrogen from the atmosphere. Again, it has to ingest or bring in a nitrogen source, most likely from protein sources. Sulfur is important because it's used in amino acids, thiamine, and biotin. Most bacteria will decompose protein for their sulfur source. Phosphorus, this is another important molecule. It's used in DNA, RNA, and ATP. It's also found in the membranes, especially the phospholipid bilayer, hence the name phospholipid bilayer. The phospho is referring to the phosphorus layer. Phosphate sources are pretty ubiquitous in the environment, and many sources are available for that. Trace elements, as I mentioned, these are inorganic elements that are required in small amounts. Usually they will act as enzyme cofactors, and they can include things such as iron, copper, and zinc. Oxygen state. We have pretty much 4 major types of states with oxygen. They're defined here, but I'm going to go to the picture. We have what's called an obligate aerobe. Obligate means it's absolute. So an obligate aerobe, what you have here is it's able to grow at the surface only because that's where the oxygen is. Facultative anaerobe is both aerobic and anaerobic growth, but you get greater growth in the presence of oxygen. Okay, so that's why you see more growth towards the surface here. Obligate anaerobe can only grow without the presence of oxygen, and that's why you see the growth down here. And then aerotolerant anaerobes, they are anaerobic. They only grow in anaerobic conditions, but oxygen will not harm them. And that's why you can see some growth up towards the surface as well, because oxygen won't harm them, but it's not required for them. Organic compounds, these are obtained from the environment, and these include vitamins, amino acids, purines, and pyrimidines. Purines and pyrimidines are the monomers that we find in nucleic acids. So looking at culture medium, this is how most microbes are going to get their nutritional sources when we are actually culturing them, is it has to be placed into the medium. And I'm not going to spend a whole lot of time on this because this is something that was defined in chapter 3 of your book when you looked at tools of microbiology. But, you know, we have chemically defined media and complex media. We've defined both of these terms already. You should be familiar with them. Again, this was chapter 3. If you forgot them already, make sure that you relearn them, okay? A reducing media, this is a media that's typically used for the cultivation of anaerobic bacteria. Reducing media will actually contain chemicals such as sodium thioglycolate that will combine oxygen to deplete it. It's heated to drive off the oxygen. Sometimes if you're growing up anaerobic cultures, you can also set up an environment that is strictly anaerobic, such as putting the petri dishes, as you can see here, in a sealed canister. The sealed canister will actually also contain a gas bag that will release, say, CO2 and hydrogen. The hydrogen will bind up any oxygen. You'll get some, water formation, but the CO2 is going to fill the canister, essentially creating an anaerobic environment within the canister. Capnophiles, these are microbes that require high CO2 conditions, and you can either use a CO2 packet or what they call a candle jar. I don't have a picture of that. One thing you want to think about is there are areas of your body where you actually have higher CO2 concentrations than the atmosphere, and one major area would be the blood. Blood on average has about 4% CO2 on the red blood cells. And if you think about that, the atmospheric CO2 is like 0.2 to 0.3%. So you have a much higher concentration of CO2 available in blood than you do, say in the atmosphere. So sometimes some bacteria actually prefer that, and that's why they are suited very well, say for bloodstream infections. But, if you were to culture it in atmospheric air, it wouldn't culture very well because there's just not enough CO2 there. So sometimes you have to mimic the appropriate conditions. Biosafety levels, you may have seen a lot of this, especially with what's been going on in the world. But when we grow cultures, we do take into account their biosafety levels. The biosafety levels are just the levels of precautions that should be taken when you're handling microorganisms. So if something is classified as BSL-1, no special precautions are taken. Basic teaching labs use BSL-1 organisms. And, when we are actually in a physical lab doing these exercises, it is considered BSL-1. BSL-2, you need to wear a lab coat, gloves, and eye protection. BSL-3, you need a biosafety cabinet to prevent airborne transmission. So now the difference between BSL-2 and BSL-3 is BSL-2 can be transmitted by direct contact. And by wearing gloves, you are actually protecting yourself. BSL-3, this is something that can be transmitted airborne. So you want to be operating with this underneath a cabinet that has negative pressure. You want to make sure you have protective PPE on, especially to sort of block any airborne transmission. That's something that we're definitely seeing with coronavirus. Coronavirus would definitely be classified as a BSL-3. BSL-4, these are really the nasty ones. Not only can they be aerosolized or transmitted in the air, but they also have very high mortality rates associated with them. And this culturing of these things needs to be handled in a BSL-4 lab, which is a completely sealed environment. You're wearing sort of the space suits with your own air supply. I think I picture that. Working in a cabinet, which is that structure they're working under in anyway, and there's always somebody there with you. You're never by yourself. There are very few BSL-4 labs in the country. Most are actually run by the federal government because of the high level of regulations. And most of the BSL-4 labs are actually kept out of cities. For example, the city of Philadelphia actually has a law on the books that states no institution or company can actually have a BSL-4 lab within the city limits. Different types of media, again, we kind of went over this in chapter 3. Selective media, this will suppress unwanted microbes and encourage desired microbes to grow. It can contain inhibitors to suppress growth. Differential media, this allows you to distinguish between one organism versus another. Some media have both selective and differential characteristics. Okay, and this is just a little summary slide that goes over the different types of media. Again, most of these were covered in chapter 3. This should just be for you to sort of refresh a little bit. Obtaining a pure culture. Last week you guys did lab exercise 4, which was the streak plate method. This is how you would actually obtain a pure culture, and the goal with this is, you initially will inoculate, then you drag out some from that initial inoculation, then you drag out some from that second inoculation. And the goal here is, you're initially putting down lots of bacteria, then you're dragging out a small subset, and then eventually you're dragging out even further small subset where you can get individual colonies. That is the goal of streaking for isolation using the streak plate method, to get individual colonies. Okay. Okay, bacterial growth. You will have one open-ended question on the exam that really comes from this chapter 6 for my CCP students, or chapter 7 for my Camden County students. And the question is to list and describe the 4-step phases of growth. Okay. Bacterial growth is primarily done by binary fission, but you can also get some budding. And essentially what binary fission is the cell will grow, it will increase in size. You need to make a copy of the single chromosome, and then eventually the cell will just split into 2. It's a very simple process. It occurs very rapidly. For example, E. coli can actually replicate about every 20 minutes. And here we can see that again. When we talk about bacterial growth, it is exponential. What that means is when you start off with 1 cell, it's going to divide, you have 2 cells. When those 2 cells divide, you have 4 cells. When those 4 cells divide, you have 8. When those 8 divide, you have 16, and this just continues. So that's what we mean by exponential growth. As I mentioned, you will have one open-ended question, and the question will be, and this is a question that's also on your lecture assignment, it's to list and describe the 4 phases of growth. Here are the 4 phases of growth, and we're going to go through each of those. There's actually a little description here for each of the 4 phases of growth. The first phase is lag phase. During lag phase, this is when the cell is introduced to a nutrient-rich environment. Because growth is a metabolic process and will occur when there's plenty of nutrients around, when the cell senses that there's plenty of nutrients, what will happen is it will actually turn on metabolic pathways to actually initiate growth. And that takes some time. So the lag phase is just sort of the quote-unquote cell waking up. The log phase is exponential logarithmic growth. Okay, this is when the cell is doubling in numbers with each division. And you'll see numbers increase during the log phase. Stationary phase is a period of equilibrium. This is when microbial deaths balance production of new cells. So essentially what you have during stationary phase is new cells are growing, but in equal numbers of cells that are dying. So you're maintaining the cell number. This is smart because you don't want to overpopulate your area, because if you end up overpopulating, you might not have enough food then to meet the demands of the cells. And then finally, we will enter into what's referred to as the death phase. The death phase is when nutrients have started to decline. As nutrients have started to decline, what happens then is you start to lose cells. And unfortunately, those cells that end up dying also now become food for the remaining cells. And those are the 4 phases of growth and a little description on each one. This is a great slide if you're sort of preparing your answer there. Okay, and that's it for Chapter 6 for my CCP students, or Chapter 7 for my Camden County students. Microbial growth, as I mentioned, this is a pretty easy chapter. Take it for what it is, and please don't spend a whole lot of time on this. My Camden County students, you guys will then be moving into metabolism, which is a very difficult topic and will take most of the week for you to prepare for. For my CCP students, you had your metabolism chapter last week, but you're going to start getting into some infections this week. Both of my schools have exams this week, so please make sure you look for the start and end date for those exams and complete your exams during those times. Okay. Have a good one, guys.